Hi, this is our first video of our biology series. So thank you so much for joining us. Remember to watch as many times as you need to. Remember to pause, rewind, take notes. So today's awesome topic, we're going to start off with DNA, deoxyribonucleic acid. And we'll also talk about protein synthesis. So let's do this, and remember, science rocks.
It is super important for you to create a biology notebook, whether it's online in Google Drive or straight up old school spiral notebook. Take notes. Take notes and draw pictures as you follow along. Copy down these 20 vocab words. They go in ABC order like this. Remember to come back later and create your own definitions of these words. Use your own words, draw pictures, or even use a funny app like ChatterPix or Shadow Puppets. So go ahead and pause this if you need to finish writing down the terms. All right, it's important to know how genetic material is organized. The genome of an organism refers to its complete genetic makeup and includes the organism's entire set of chromosomes. The total number of chromosomes in a normal human is 46. 23 chromosomes from the mother and 23 from the father. So genetic information is encoded in DNA. So you see this double helix right here. DNA is located in the chromosomes of the cell. So you see it all stretched out to show you the DNA double helix. A section of a chromosome that controls a particular trait is a gene. So genes are a kind of like a blueprint for an organism. There are 4 nitrogenous bases in DNA. There's adenine, thymine, guanine, and cytosine. The A is always with the T, unless there's a mutation, and the C should be with the G. I always think of it as the stick letters, so an A with a T, or it makes a word, at. It could be T with an A, vice versa. But the stick letters always go together, and then the curvy letters, C and G, are always together. C and G or G with C. Notice down the backbones of this double helix picture. These are the backbones, the sides. There's the sugars, which is the D for deoxyribose, and then there's the phosphates, which are in yellow, the Ps. They alternate down the backbones of the DNA double helix. The deoxyribose sugars bond directly with the nitrogenous base, and then the phosphates are in between. Notice the A with the T, and they're actually bonded together by weak hydrogen bonds. Up here you can notice the G with the C, and they're bonded together by the weak hydrogen bonds. So before a cell undergoes cell division, it will want to replicate its double helix DNA. So I'm going to go ahead and unwind this so we can look at an example. Say there's an A paired with a T, a C paired with a G, and maybe a T paired with an A. An enzyme is going to come along and unzip these weak hydrogen bonds that are holding them together. And then you'll have these 2 strands that will separate from each other. Okay, they're going to pull apart from each other. So I'm going to go ahead and rewrite those bases so I can show you. So we've got a TGA from this right side, and then the left side we have an ACT. When the strands separate, there's free-floating nitrogenous bases in the nucleus, we're inside the nucleus, that will now pair up with these strands. And the sugars and phosphates will be attached as well to create the new backbones. So they'll attach to the correct base. So in the nucleus, there's going to be an adenine that wants to attach to this thymine. There's going to be a cytosine that wants to attach to the guanine, and a thymine to attach to the adenine. And also in the next one, you're going to find a thymine in the nucleus to attach to the adenine, a guanine to attach to the cytosine, and an adenine to attach to the thymine. They will twist back up into their double helix form. And now notice you have two identical strands of DNA. DNA replication. So now both new cells will know what to do. Protein synthesis. Genes are used by the cell to make proteins. In order to produce a protein, genes must be transcribed and translated. So you take one of the strands of DNA from the double helix, and it goes through something called transcription in the nucleus, where it copies into a messenger or mRNA molecule. Notice it's one strand, and the bases are paired the same, except there is no thymine in RNA, so there's no T's. It's replaced with uracil, so a U. So notice you'll see these U's in the RNA strand. So if you have a C in DNA, you can pair it up with a G in RNA. If you have a T in DNA, you can still pair it up with an A in RNA. If you have a G in DNA, you can still pair it up with a C in RNA. Now, if you have an A, here's the difference. In DNA, you're going to have to pair it up with a U in RNA. Okay, there's no T's, there's no thymine in RNA. So after that, the mRNA moves into the cell's cytoplasm and attaches to a ribosome where it is translated into a protein. A little bit more about transcription. There's three main processes that occur. And the first step is called initiation. So during initiation, enzymes bind to a DNA sequence and unwind. See how it's starting to unwind the double helix to expose the strand of nucleotides. Now, when I talk about nucleotides, a nucleotide is a nitrogenous base, an A, T, C, or G. So say you've got a C, along with the deoxyribose sugar and the phosphate. So it's the grouping of those three things when I talk about nucleotides. The second step is called elongation. So go ahead and write down the second step. As the DNA molecule unwinds, an enzyme called RNA polymerase pairs the RNA nucleotides with the DNA nucleotides. So, for example, here, and it starts on the left, if the DNA strand reads CTG, right here, the first three, CTG, the complementary RNA strand would read GAC. Okay, and you'll group them in threes. Next, on the DNA, you can see ACG and UGC would be its complementary end RNA. Okay, next in the DNA, you see GAT with CUA end RNA. So it's going to go through and match up the RNA with the one side of the DNA strand. The third step is called termination. Once the gene is transcribed, the new RNA molecule, so this right here, breaks away and the DNA strands are wound back together. Translation. I always remember transcription happens before translation because it's ABC order. Transcription comes before translation. So here are the steps of translation. Number one, a ribosome attaches to this mRNA strand. So we've left the nucleus, we're in the cytoplasm, and we've found a ribosome. Each 3-base segment of the mRNA strand is called a codon. So say you have a U, a C, and a G. Those 3 together would be referred to as a codon. Step 2, transfer RNA molecules carrying these amino acids approach the ribosome, and it goes from the 5 to 3 direction. The tRNA molecule, step 3, whose anticodon corresponds to the first codon on the mRNA strand, quickly attaches. So here's the codon on the mRNA and the anticodon of this tRNA that matches up, and they match up just like our base pairs do. Remember to use U's instead of T's since we're both dealing with RNA molecules. Step 4, a new tRNA molecule carrying another amino acid attaches to the next codon on the mRNA strand. Here's its anticodon, and here's its amino acid. Step 5, as amino acids are added next to each other, peptide bonds form between them. And 6, previous tRNA molecules detach from the mRNA strand and they depart from the ribosome. Notice they left their amino acids behind. So, step 7, the chain of amino acids continue to grow until the ribosome reaches something called a stop codon at the end of the mRNA strand. This signals that no more amino acids should be added. The result is a polypeptide, and it will fold into a functional protein. How exactly does a sequence of nucleotides result in a chain of specific amino acids? Well, the answer lies in the genetic code in the codon table, which determines which amino acids correspond with these 3 base codons. To determine which amino acid corresponds to a codon, find the row matching the first RNA base, the column matching the second base, and then the specific codon containing the third base. So if your RNA is UAC, you'll look for the first position of U, second A, now you know you're in here, and the amino acid UAC would be abbreviated TYR. Another example, say your mRNA is GUG. Your first position would be G, second U, so now you know you're down here. GUG, the amino acid would be abbreviated as VAL. An mRNA transcript must have distinct starting and ending points, which are indicated by a start codon of AUG, which is this abbreviation MET amino acid, and a stop codon. Could be UAA, UAG, or UGA. You'll find these 3 different stop codons. The final structure of a protein is determined by the sequence of its amino acids. Once a chain of amino acids is produced, it undergoes a series of folds, bends, and twists to arrive at its final structure. And there are 4 distinct levels of protein structure. Come back later and watch these additional videos and take more notes, or pause and do this now. All right, so for some practice questions. Number 1, cells store genetic information in DNA. That genetic information is used to synthesize what? Is it A, polysaccharides, B, carbohydrates, C, proteins, or D, genes? Why don't you pause this and think about it on your own? Okay, so the genetic information stored in cells is used to synthesize or make proteins. So, C. Remember, during protein synthesis, DNA is first replicated to form other copies of DNA. Then it is transcribed to form strands of RNA. And finally, the RNA is translated into proteins, which help the body perform numerous functions. So just by process of elimination, we haven't talked much about polysaccharides or carbohydrates. And to synthesize means to make, and the cell makes proteins, not genes. Number 2, in which of the following ways does genetic information typically flow in an organism? Is it A, from RNA to proteins to DNA, B, from DNA to proteins to RNA, C, from DNA to RNA to proteins, or D, from proteins to RNA to DNA? So pause this and think about it on your own. Heredity information is passed from parents to their offspring in the form of genes. Genes are segments of DNA that carry specific traits. So in general, genetic information flows in the following manner: DNA to RNA, and then finally to proteins. So C. Remember, DNA is transcribed into RNA molecules. Then the RNA molecules are translated into proteins. If we remember that the ultimate goal is a protein, then that only leaves us with C. Number 3, the table below shows the codons that make up the genetic code in the sequence of nucleotides that corresponds to them. To determine which amino acid corresponds to a codon, find the row matching the first RNA nucleotide, the column matching the second nucleotide, and the specific codon containing the third nucleotide. What sequence of amino acids would be coded by the following set of nucleotides? We've got AUG, CCU, ACG, UGG, and GAC. So go ahead and pause this and think about it on your own. So if we look at this, we will first look at AUG. You're going to find the A on the left in the first position, the U up top in the second position. So now you know you're in here. And AUG lines up with the amino acid abbreviated MET. And if you remember, that is your start codon. So by process of elimination, we can mark off B and C, okay, because A and D both start with MET. All right, next let's look at CCU. So find the C on the first position on the left, the C up top in second position, and now you know you're in here, and CCU matches up with the amino acid abbreviated PRO right here. Okay, so I know that A is going to be the answer. I'd probably go through and check all of the others just in case. Number 4, a sequence of nucleotides is shown below. What does this sequence provided code for? A, lipids, B, carbohydrates, C, ATP, or D, proteins? So pause this and think about it. A sequence of nucleotides in DNA ultimately codes for proteins, y'all. So D. Number 5, a codon is a set of 3 nucleotides that correspond to a specific amino acid. The table shows various DNA codons corresponding to its amino acid. 2 strands of DNA are identical except for one codon. As a result, they code for slightly different proteins. Based on the information in the table, which of the following statements could be true? So I've gone ahead and color-coded these so I didn't have to find them. A, one strand of CGC codon instead of CGG. Now, look at that in red right here. Even though the codons are different, they both actually code for the same amino acid. So A is not going to be it. B, one strand contains CAC codon instead of CTC. Now, those actually code for different amino acids. So it's going to be B. If you go through and look at C, CCC and CCA code for the same amino acid, so that one would be fine. And ACG and ACA also code for the same amino acid, so that would be fine. Okay, after fully covering the topics of DNA and protein synthesis, you should be able to explain how molecules called DNA serve as the instructions that all living organisms use to produce proteins, which in turn perform essential functions of life. So, I challenge you to create a project, a presentation, an experiment, something, and show your teacher that you can explain this.